10 introns. DNA found in nucleus. Then internal structures, no membrane-bound organelles are present in prokaryotes, while eukaryotes have membrane-bound organelles. Ribosomes are 70s ribosomes, while eukaryotes have 80s ribosomes. Reproduction is asexual, binary fission. DNA is singular, haploid, while in case of eukaryotes, it's asexual, that is mitosis, or sexual, that is meiosis. Then DNA is usually paired, diploid, or more. Average size is 1 to 5 micron, and here it is 10 to 100. Then you can also understand the graphical representation of the difference between prokaryotes, as I have mentioned. But the difference here is, here you can also see the biological functions of the each organelle. So it will give you better insight. It's so all these slides what I have presented, if instead of those slides, if you go for just this single figure, you can easily get the knowledge of difference between prokaryotes and eukaryotes, the biological functions of the cell organelles also, or components of the cells. Then I'm coming to each cell organelle one by one. Here, let's say plasma membrane, that is here, this one. Then these components I have explained clearly. So I just wanted to introduce some of the things, but most important thing, what I want to introduce or give you some idea, for example, flagella or cilia, how it could look like. Then here microfilaments, intermediate filaments, and microtubules compose a cell's cytoskeleton, which maintains the cell structures in proper shape. Now, endomembrane system like rough ER, endoplasmic reticulum, and smooth endoplasmic reticulum. Instead of reading the detailed text, you just need to understand, see this figure where you can see the dots, that is ribosomes, and dot-free structure is no ribosome present. So this is rough endoplasmic reticulum. This is a smooth endoplasmic reticulum. Here, as a layman, you can just understand the function here, protein synthesis, here lipid synthesis. And the same thing you can see, they have ribosomes attached on their surfaces, while smooth do not have ribosomes attached on the surfaces. It manufactures proteins and transports them to various parts, while SER, smooth, manufactures lipids and transports them to various parts. So this is basic clear difference between rough and smooth endoplasmic reticulum. What are the structures in details inside of endoplasmic reticulum? Like endoplasmic reticulum, here you can see chromatin, nucleoplasm, nuclear pore, nuclear envelope. And here, what are these things here you are saying? This is basically nucleus. And what you are saying here, this is rough endoplasmic reticulum, which is basically connecting to the nucleus. So the transfer of proteins, different things could be transferred to different places. Nucleoplasm is just like cytoplasm, where cytoplasm is the liquid part of the cell inside. Similarly, nucleoplasm is present inside the nucleus. Now, there are different definitions for all these components, and you can read these components. What is nuclear envelope? You can just understand similar to cell membrane. And then what are their roles? And then what is endoplasmic reticulum? What is the purpose of rough and smooth endoplasmic reticulum? What is the real function and how it transports the synthesized materials, proteins, or lipids? Where rough RER manufactures proteins and then transports to the various part. Similarly, SER manufactures lipids, transports it to various parts. And then there is another important structure, that is Golgi apparatus. If you want to understand how Golgi apparatus could look like. Then this is here, the yellow color. This is indicating Golgi apparatus, cis phase and trans phase. That is simple definition. Like Golgi apparatus is called cells post office, and these are some of the most common exams for any competitive exams. So Golgi apparatus is an organelle that modifies, sorts, and transports lipids, proteins, and other macromolecules to different parts. So this is called a cells post office. This is what you need to remember. Then it comes to lysosomes. Then lysosome is very important cell organelle. And here you can see what it says. Lysosomes are the cell's garbage disposal. And these are the terms which you need to remember if you are writing some competitive exams. Then digestive enzymes within the lysosomes aid the breakdown of proteins, polysaccharides, lipids, nucleic acids, and even worn out organelles. In single cell eukaryotes, lysosomes are important for digestion of the food they ingest and recycling of the organelles. These enzymes are active as a much lower pH, at a much lower pH. And these components are so important for a cell so that everywhere there comes the questions in the exam. So you need to remember these things. So please remember what are the terms, for example, cis phase, trans phase, lumen, cisternae, lysosome, what is lysosome, what it is called, what are the other names of lysosomes, why it is called garbage disposal, why Golgi apparatus is called cells post office. So these are some of the terms which you need to remember. And these are so attractive words that if you are revising or passing through some of the text or videos, you will easily remember it. Then, in order to present the entire endomembrane systems, here this is the figure which represents the entire endomembrane systems, which works to modify, package, and transports lipids and proteins. And that is here in this figure. Now it comes, if you want to see lysosomes, how it looks like in terms of a structure, why it is so important, then there are some research paper, but here you don't need to understand the research paper. You just need to remember this one-line sentence. Lysosomes are the organelles responsible for breakdown cellular waste. They are filled with digestive enzymes. Lysozymes have a low pH on the inside, that is acidic, that allows the digestive system to work. Now, there is another organelle, or sorry, a cell, which is showing the real role of lysosome, and it is also one of these cell which works very similar to the role of lysosome, and it is very famous for saving the human from different kind of infections. So a macrophage has phagocytized a potentially pathogenic bacterium into a vesicle, which then fuses with lysosome. Here you can see the role of lysosome within the cell so that pathogens could be destroyed. And other organelles are present in the cell, but for simplicity, here what you are seeing is lysosome, bacterium, vesicle, and the process of phagocytosis. And that is how this immune cell or macrophage works. Now it comes, ribosomes are the cellular structures responsible for protein synthesis, because we have described rough and smooth endoplasmic reticulum. Now it comes to ribosome. So we know what ribosomes are doing. So it is responsible for protein synthesis. And we also know that 70s RNA is present in eukaryote and 80s RNA ribosomes are present in prokaryotes. So how the structure in general are depicted to represent the ribosomes, that is here. What you can see, the small unit and large subunit. Here, the left side is for prokaryote, that is 50s plus 30s, and here it is 70s. This is called 70s RNA. And then 60s plus 40s unit, here it is called 80s ribosome. So there are something, I'm not going too much deep, but how the proteins are synthesized, and this is how you can see the representative figure, E site, P site, A site are present, and then the codons are passing through how the synthesis of proteins could be taking place in a ribosome or bioribosome. Then it comes to mitochondria, which is very important, and it is called the powerhouse of the cells. So mitochondria are eukaryotic organelles that make chemical energy via aerobic cellular respiration. And how a mitochondria could look like. And here, membrane, outer and inner membrane, ribosomes, intermediate space, matrix, mitochondrial DNA, and Christie. That is why there are a number of places where you can see it is written as mitochondrial genes, and you can see mitochondrial DNA is present. Thus, definitely mitochondrial genes are also present, and they have specific roles. Now, peroxisomes are small round organelles enclosed by single membranes. They carry out oxidation reactions that break down fatty acids and amino acids. They are also detoxify many poisons that may enter the body. Alcohol is detoxified by peroxisomes in a liver cell, and by a byproduct by these oxidation reactions is hydrogen peroxide, which is contained within the peroxisomes to prevent the chemical from causing damage to cellular components outside the organelle. So peroxisomes are multifunctionals, and their sizes are 1.0 micrometers, normally 0.1 to 1.0 micrometers, which contains a dense crystalline core of oxidative enzymes, and it has more than 50 enzymes involved in diverse activities. These organelles were so named because the site of synthesis and degradation of hydrogen peroxide, a highly reactive and toxic oxidizing agent. So thus, the structure of the peroxisome could be seen here. They are particles of about 100 to 500. There are different sizes mentioned, but what you need to understand the function of peroxisome. A lipid bilayer membrane surrounds which regulates what enters and exits peroxisomes inside a dense matrix urate oxidase crystalline core. Then the difference, if you want to understand, since there are some functional similarities between lysosomes and peroxisomes, you can understand here this represents the difference between lysosomes and peroxisomes, membrane-bound sacs performing a digestive function, membrane-bound sacs, peroxisomes performing digestive function. This is similar. Contains enzymes to digest food wastes, invading bacteria, and break down old organelles. Here, enzymes in peroxisomes are oxidases that catalyzes redox reactions present in animal cells only. Here, they are contains many peroxisomes to break down alcohol. And again, Golgi apparatus produces lysosomes here formed by budding off from ER. Tray Sachs diseases present in animal cells only. Now, the next class on for the cell and the biology class will be on animal cells versus plant cells, and more things will be covering.